Hey everyone, welcome to Professor Long's Lectures in Microbiology. I'm Professor Bob Long. As you know, these videos are intended to provide a solid foundational understanding of some of the fundamental principles in microbiology. If you are taking this course with another instructor, please make sure that you learn all the material and all the details that that instructor requires. Again, this is for a solid foundational understanding of some fundamental principles in which we can add details later. The amount of details that are added by each instructor may vary. So, we've been talking about microbial growth. We've done microbial metabolism and microbial growth. I hope you've watched those videos. If not, you should look them up. We're continuing a little bit with microbial growth as far as the chapter in your textbook. We need to finish up, how do we measure microbial growth? We've talked about things that can affect microbial growth, like oxygen concentrations, whether they're aerobic and anaerobic, temperature, pH, and a number of other factors, nutritional requirements. We need to talk about how do we measure microbial growth so that we can get microbial counts. One of the things that we know is that we don't measure microbial growth by the size of the cell. You know, microbes really most often, especially bacteria, are not like humans where we start 28, 29 inches long and we can end up as much as 6.5 feet tall or taller. Most bacteria are roughly the same size within a specific genus and species. Like all E. coli are roughly the same size as other E. coli. Same thing for Bacillus subtilis or Staphylococcus epidermidis or all these others. The way that we measure bacterial growth is we measure the number of cells. How rapidly are they growing? And we've talked about how bacteria divide by binary fission and how their growth is rather exponential, especially during what we call the log phase of growth, where 1 becomes 2, and then 4, and then 8, then 16, and they double and double and double, to where within about a 24-hour period, we could have somewhere between 16 and 17 million bacteria growing. It's a huge number in a short period of time. So that's how we kind of measure the bacterial growth, is by the number of cells present, not by the size of the organism. Okay? There's a number of ways that we can measure bacterial growth, and some of them are called indirect methods, in which we can't count all the bacteria present, so what we do is we count a small sample and then we do calculations and estimations of how many are total. For example, if I have a 10,000 gallon container in an industrial situation where I'm producing something and there may be some bacteria present, I can't count all of those bacteria, but what I can do is siphon off some, do a count there, and then estimate out. For example, if I have 10,000 gallons, I might measure how many are in a particular gallon and then multiply that by 10,000. That's a simplistic view of how it would be done. It's not really exactly how it's done, but it gives you an idea of the concept. So we're going to talk about some of the indirect and the direct methods of measurement and talk just a little bit of detail, a brief overview of each one. Excuse me. So we know that when we count bacteria, we're counting the number of cells. And in direct counts, you actually count cells, or you count what are called colony-forming units, or the number of colonies. But really what we want to get down to is the number of cells. And in indirect methods is sort of where we count a small population and we do some calculation to the total amount in a particular volume of fluid or over a surface area of a solid. And we can record them as number of cells per milliliter of fluid, or the number of cells per gram of solid if we were looking at some kind of solid material. And again, as you know, bacteria grow in such high numbers, we really can't count them all. So we have to do some kind of estimation or do a smaller measurement and then calculate to where we would think is a good estimation of the total number of bacteria. And most of these methods have proven to be pretty accurate for the most part. Now, if we want to talk about one method of measuring bacterial growth, one of the things that we can measure is the turbidity of bacteria. And this marker is not writing very well, so I should get rid of it. But we can measure turbidity. Now, we've talked about turbidity as a type of growth characteristics of bacteria where they produce a cloudiness when grown in a broth. And so when we measure turbidity, essentially what we are measuring is how much light can pass through the medium. And so you use, you take a light source that gives off a known amount of illumination. You pass it through a tube of medium that has a bunch of bacteria growing in it, and as the light passes through here, it's collected by something called a spectrophotometer. Or some people like to say, some of my classmates when I was a student would say spectrophotometer, because it's going to meter or measure the light passing through. And essentially, as light passes through a medium, only a certain amount of light would make it through. And the more turbid the medium, increased turbidity means decreased amount of light passing through. And by the way, I was watching one of my previous videos just in a short segment, and I realized I spelled light, L-I-H-G-T. If you saw that video, please don't think I'm an idiot. I know how to spell. Just when you're teaching, you're thinking about where you're going, not where you're at. As one of my stats professors used to say, sometimes I say 1, I mean 2, and I write 3, because we're thinking on our feet, so to speak. But you can measure the turbidity of growth, and the more turbid it is, the less light would pass through, and the more turbid it is means the higher number of bacteria. So increased turbidity can also mean an increase in the number of bacteria, or whatever microbe you're measuring. We're focusing a lot on bacteria. Now, one of the things I want to point out about this is this does not allow us to measure very small changes. So if I were to go, say, from a handful of bacteria, say 1,000 to 1,000,000, I could measure that difference. You know, 1,000 would let a lot of light through, would not be very turbid, and a very turbid medium would mean, you know, lots of bacteria. But if I went, say, from 10,000 to 12,000, we would not be able to really measure that amount. So it's not always the best way to measure the amount of bacteria, or at least it doesn't give us direct measurements of the number of cells. It can just give us an indication if there's more cells or less cells. So not always the best way, okay? Now, another way that we can measure the number of bacteria is by measuring their metabolic activity, okay? And again, these are broad overviews. I'm not going to go into extreme detail, but we can measure what's called the metabolic activity of an organism. Let me grab another marker. I know this one writes really well over here. So we can measure metabolic activity. Of course, in a chemical reaction in metabolism, we're going to take substrates and turn them into products. And so we can measure how fast they burn up the substrate or how much product is produced. So there's a couple of things that we can check here is that we can measure the amount of product or how rapid the product is produced. So when we have a chemical reaction, essentially what's happening is, for example, if I have some sugar and I have a fermenting bacterium, it's going to ferment that sugar sometimes into an acid. So I can measure the acidity of a solution. And if I start with something that has, say, a pH of about 7.4, and I know over a given amount of time that a certain number of bacteria can produce so much acid from a certain amount of sugar, then if I have bacterium A, this is my abbreviation for bacteria, as you guys might know, the beta symbol, but bacteria A might produce a certain amount of acid. Bacterium B might produce acid faster. So in a certain amount of time, let's say over a 24-hour period, I can measure the pH of a solution as it's changing as the bacteria are fermenting acid. And if one produces acid faster, then I might assume that this one has a higher number. Or actually, the better way to do it is if I have 2 tubes, and in tube A, I know I have a certain number of bacteria producing a certain amount of acid as a rate, and if sample B produces acid faster, there must be more bacteria present if they are doing it in the same amount of time. So we can measure the amount of product produced. Another one thing that can be measured is we can measure the rate at which substrates are consumed. Because we do understand a lot about bacterial metabolism, then I can measure how fast the substrates disappear. It's just kind of like taking someone to a buffet. If you only take 2 friends, how long is it going to take for all the food to disappear? Or if you take a whole, football team, how long is it going to take for the food to disappear? And the faster the rate, the more bacteria are present. Okay, so we can measure the rate at which substrates are consumed. A couple of the substrates that we can look at and measure is we can measure like glucose or sugar consumption, and there are indicators for this. And we can measure oxygen consumption sometimes. How fast do the bacteria utilize all the consumption or consume all the oxygen in their metabolic pathways? And so that can give us an idea. If I have the same bacterium in 2 cultures, and one is consuming the products very slowly and the other is consuming it more rapidly, I can assume that one has a much higher number of bacteria or microbes present. And finally, a third thing that we can measure is the rate of reduction of certain dyes. So, if I know there's a certain concentration of a dye that gives an indication, for example, one of the dyes we can look at that is used is methylene blue. And at a certain concentration, we have a certain color. As it's broken down, it becomes more colorless or loses color. I can't write. So, I can measure the rate of the reduction of methylene blue over a period of time for 2 different cultures, and if I have the same bacteria in 2 or more cultures, then the faster the blue disappears, I can assume the more cells that there are. Again, these aren't always the best way to measure the number of cells, but it can tell us the abundance of them to some degree. Now, there's more direct ways that we can measure bacterial growth. So, these are somewhat indirect methods. They don't always tell us the exact number of cells, but we can measure in direct ways. One of the things that we can do is we can actually do what are called direct counts, where you can actually count the number of cells present. One way to use is you use a machine called a coulter, after the doctor who invented it, C-O-U-L-T-E-R. A coulter counter. And the coulter counter method basically uses an electrical signal. Oops, God, I can't write today. Guys, please forgive me. But it uses an electrical signal to count cells. And essentially, if I have a fluid passing through something and I have an electrical signal across here, every time something interrupts that signal, then I have count that as a cell. So I can pass a number of cells through an electrical signal, and the computer will actually count the number of disruptions or interruptions. Now, it's a real good way to count almost the direct number of cells. One of the problems is it works best if only the bacterium of interest is present. So if I'm trying to count the number of, say, E. coli in a sample, and the sample is contaminated, I'm going to get a cell count, but I may not get the number of E. coli. So, you got to make sure you have a pure sample. and there's no contamination. And also, it can count dead cells. So whether the cell is alive or not, if it passes through, it's going to count it. So one of the things that we know about it is this does not always give what we call a viable cell count. Viable meaning live cell count. So we're not always counting live cells, okay? But it is a really good way to count the number of bacteria, and if you know your sample's pure and you've eliminated any of the dead cells, then you can get a really, a direct count of the number of bacteria, okay? Now, another direct method of doing it is what's called a plate count. And, personally, I don't always necessarily think of this as a direct method. It's partially direct, but it's considered one. But you can do what's called a plate count. And what you do is you use a thing called a Quebec counter, okay? Or a Quebec grid. Now, and very often referred to as a Quebec colony counter, because what you're measuring is you're counting the number of colonies that are present. And essentially what happens is you might have a plate that has tons of bacteria growing all over it in little colonies. And you put this on a grid, and then you can count a specific area. And what we're trying to guarantee is that we've uniformly spread the bacteria out, and we can see the certain colony forming units. Not every colony is an individual cell, but if we spread them properly and we hopefully get down to one or a couple of cells, then as it grows a colony, then the number of colonies growing should represent the number of cells that I started with. And so, and as you count a specific grid, then you can estimate out to the total surface area how many bacteria are present. Another method of doing a count, so you need to know that plate counts are sort of a way to directly count, but you still do an estimation. You use the Quebec colony counter, count a specific area, and then estimate out towards the total surface area. And you are somewhat directly observing the cells. It's really good at measuring viable cells because dead cells won't form colonies. Now, another way to do it is through filtration. And essentially what happens is you pass air or liquid through a filter. And what we know is that the filter will have pores smaller than the microbe of interest or the bacterium of interest. So as we pass the bacteria through the filter, all the bacteria will get trapped on the surface of the filter. Then we resuspend in a medium, and we can count. And this will give us an idea of how many, and of course, you have to resuspend in a medium, you have to inoculate something, and you have to grow. And the number of colonies that form should be the number of cells that were present. And so again, we can measure what are called the colony forming units. So the filtration method is a really good way of isolating the cells and then growing those that are alive and getting sort of a direct count. Now, one of the things that we need to talk about, and I'm not going to go into super detail on it, but I want you to be aware of the technique. We're not going to do any major calculations, but sometimes in order to do counts, we have to do what's called a serial dilution. Now, in serial dilution, you're going to dilute something. You're going to put it in increasing amounts of liquid so that it becomes more dilute. Something like taking a glass of milk and drinking it, and then you mix that with half a glass of milk with half a glass of water. It's going to taste less milky. And then if I do that again, I take a half a glass of that and put it in a half a glass of water. I essentially have about 25% of my solution is milk, and if I do it again, then I get 12.5, and again, and I keep going down and down and down, so I can dilute something in a series of dilutions. That's all that serial dilution means. And essentially what happens is this. If I have, let's say I take 1 milliliter of something, and inside that 1 milliliter, let's say I have 1,000,000 bacteria. If I took that milliliter and I tried to put it on a plate and count it, with 1,000,000 bacteria, my plate would be overgrown. And it's real hard to count 1,000,000 things. So it takes a while. But what if I took this solution and I take my 1 milliliter and I place it into 9 milliliters of sterile broth or a sterile solution? So now, instead of having, I'm starting with 1,000,000 bacteria per milliliter. If I put that 1,000,000 into 9 more milliliters, now then I can say I have 1,000,000 bacteria per 10 milliliters. And if I divide out 10 for both of these numbers, I'm essentially going to get down to 1 milliliter, and I take a 0 off. So now I have 100,000 bacteria per milliliter. If I plate that out, my plate would still be overgrown. But what if I take these 100,000 in 1 milliliter? I take 1 milliliter of this solution, they're equally distributed, and I place that in 9 milliliters of sterile solution again. Well, essentially I'm dividing everything out. Now I have 100,000 in 10 milliliters, or I could say that I have 10,000 per 1 milliliter. So in a series of dilutions, I'm decreasing the number of bacteria. And if I did this again, I put 10,000 into 10 milliliters, then I essentially have 1,000 per milliliter. And I could get down to 100 per milliliter or 10 per milliliter, and theoretically I could get down to 1 per milliliter. Through a series of diluting out the number of bacteria in a solution, I can get to a small enough amount that if I plated that out, I could actually count all the colonies. Now this really works kind of in reverse. I don't always know the number of colonies I have in a milliliter. But what I can do is I can dilute them out and plate it. And if it's overgrown, I dilute it again and plate it, and I dilute it again until I get down to some number of dilutions where I know that I'm down to, say, maybe 100 or 10 colonies or a certain number of colonies. Then I can calculate backwards to figure out how many were in the original solution. We call those serial dilutions. Okay. Now listen, this is not a really complete explanation of all the ways that we can count bacteria, or even the best ways to count them. And it's not even a thorough description of how each is done in great detail. But I want you to be aware of these methods. So I want you to be aware that we can measure the rate of metabolic activity by measuring product production, measuring substrate consumption, or measuring sort of the like if they produce gas or something, or an acid. We can also measure things like turbidity. Again, not the best way. We can do direct counts, we can do plate counts, and we can do filtration methods, and then we can do serial dilutions and work our way backwards to figure out the number of bacteria present. All these techniques can be used in a variety of situations, whether we're looking at the number of bacteria that we're taking from a sample from a wound, or you can do it, really a lot of this is done in industrial applications when we're looking at the amount of contamination in a volume of liquid or product that we're producing. If you're producing a particular solid product, like maybe some grain or what's the word I'm looking for? Some soil, and you're trying to measure the number of bacteria present, then you can do this in a number of ways. These are some of it's a basic overview of some of the methods that we can kind of figure out how many bacteria are present in something, because some bacteria are not harmful in small numbers, but as the number grows, they can start to become harmful, or they can start to disrupt whatever it is that we're trying to use them for. So too many bacteria in a particular feed might cause cattle to get sick, and so on and so forth. Or measuring the number of bacteria in milk. You know, when you buy certain food products, dairy products in particular, they're not sterile. They're not completely absent of microbes. Most of the microbes present are not harmful, and they're in such small numbers due to processes like pasteurization and other things, so that they don't do harm. But then as the bacteria grow over a period of time, they can produce spoilage, and some of the waste products can make you sick. That's why we have expiration dates on foods. That's why we do things to control microbial growth like refrigeration and other things, preservatives, and we're going to talk about that in the next video. So hopefully you guys understand a little bit about microbial metabolism so that we can understand how we can evaluate which bacteria are present by growing them on different types of media and looking at some of the growth characteristics. That's excellent in trying to identify which pathogen is causing a disease or which particular microbe is causing spoilage. Then we can measure the number of bacteria present, and then we can use all of that information to sort of either preserve foods, prevent infection, prevent spoilage and things. What we're going to look at in the next video is we're actually going to talk about how we control microbial growth to prevent those types of things, to prevent infection, to provide antisepsis or for disinfecting and for sterilizing and preserving foods. Okay. Again, this is sort of a brief overview. I know it's not probably the best explanation in great detail, but that's not the purpose of this course for this particular topic. So I hope you learned something. I hope it's a little bit understandable to you, and I hope you had as much fun as I did. If you like the video and find it helpful, please hit like and subscribe, or leave me a comment, and hope to see you in the next video. Thanks for watching.